Today I'm going to be talking to you about our GeneArt gene synthesis service. So in the gene synthesis workflow, this is really where the focus is for our GeneArt gene synthesis service offering. We go through gene optimization, gene synthesis, subcloning, and plasmid production are really going to be what fits into our offering. And all of this is made possible through our next generation gene synthesis platform. So we have a full bioinformatics suite that helps with the design as well as optimization of your gene. We then synthesize and assemble that gene for you. We will clone it, provide sequencing and final documentation for both your gene that's been produced, and if you do also require plasmid preparation, we have full final documentation for the plasmid as well. At the core of this is our GeneArt Gene Optimizer software. Our GeneArt Gene Optimizer software takes into account over 20 different parameters to provide an optimized gene to provide maximal expression in your particular system. And that goes beyond taking into account any codon usage tables. That includes looking at GC content, looking at any unwanted motifs or any potential secondary structures that could be created based on the sequence. To give you a little bit more detail on how this process works, when we're thinking about a wild-type gene, that gene is really, the sequence there is really more designed around optimizing things for the cell with whatever the cell has been evolved to use for that particular protein. It's not really designed around providing optimal expression. So what our software is really focused on is how do we provide you the highest level of expression without introducing any unwanted motifs or secondary structures. And so it's really trying to find that trade-off. So if we look at this example here with this amino acid sequence that spells out LifeTech, we have in the wild type, we may have some less than optimal codon usage, and then we have an idea on kind of what the best codons would be for our particular host system. But we can't make all these green necessarily because that could create repetitions, it could create unwanted motifs, might create secondary structure that would also affect how well the gene is actually expressed. And so what our software does, we use a sliding window where we can go and optimize a particular codon at a particular site, but then also look at what's going on around that particular site and come up with more or less an optimal set of codons that give us the best expression within the system. This isn't just something that we've worked out in theory. This is something that we know works. Here's some data that is from a peer-reviewed publication. So as you can see, our gene optimizer algorithm performs very well and performs very consistently across these different scenarios that you see here. And in many cases, actually outperforms a lot of the competitors that are on the market. In addition to just synthesizing your gene, we are able also to provide subcloning services. And that basically comes in two flavors. So there's one option with our express cloning offering where we have a set of Invitrogen vectors that we can basically provide your gene directly in that vector. So you can order your gene from us and receive back a ready-to-express clone. And this system is also adaptable. So if you are in need of a fast turnaround scenario system and you have a vector that you use very frequently, we can actually onboard your vector into this express cloning process. We do also have our gene synthesis plus subcloning, so the more traditional subcloning offering. This is very flexible, so we are able to do this into our suite of over 150 Invitrogen vectors, as well as we can do this into most customer vectors as well. This process does take a bit longer, but is our most flexible offering there as far as providing your gene back to you in the vector of interest. And so with our standard service offering, we will deliver about 5 micrograms of the vector of the clonal vector. We are also able to do scale up through our plasmid preparation services. And we are able to go from anywhere from a MIDI prep all the way up to multiple milligrams, so 20 mgs of DNA if that's what you need for a downstream process. This comes with a full set of QC documentation, and we're able to offer this in TE, water, or PBS. If you do need TSC-free production, we are able to offer that as well. And on this slide, this is just a kind of a high level of what our turnaround times look like. We are able to provide things in as little as 7 business days in our standard offering, or as little as 5 business days with our super speed expedited service. And then, for example, with express cloning, we look at kind of where our gene size is, it's about 2 days, and our sub-cloning typically adds about a week onto these. There are 3 ways that you can place an order with us. So there's offline, which is reaching out to our customer care team or reaching out to your local representative. You can also order online. We have a set of portals that you can use to actually place your order and have it go directly into production. And then we also have an API system. So if you do have a need for an automated ordering process or additional support with regard to automated design support as well, we do have an API which can be used by a programmer essentially to be able to help you with the design and then ordering in a more programmatic method. Part of our commitment to making the world healthier, cleaner, and safer is our commitment to zero waste. So our Regensburg facility, where all of our GeneArt services are performed, is a certified zero waste facility, which means that 0% of the waste that we produce at that facility actually goes to landfill. So if you'd like to learn more, get in contact with us, you can go to thermofisher.com/GeneArt. Feel free to contact your local sales representative. If you're in the state of Massachusetts, that would be me. And then you can also email our team at geneartsupport@thermofisher.com. Thank you very much for your attention.